We are moving to the last talk of this session, which is also very exciting. So we are going to have Dmytro Fishman. Just to say a few words about him, Dmytro is a lecturer in artificial intelligence at the Institute of Computer Science at the University of Tartu in Estonia. He is also the co-founder and chief science officer of Better Machine, a medical imaging startup that is building intelligent tools to enable doctors to diagnose cancer more efficiently. And in his talk, he's going to talk about this interplay of taking state-of-the-art models and putting them into sort of working system, which I think is a very exciting topic, and I'm really excited to hear what Dmytro is going to say. Dmytro, we are very pleased to have you here today. This stage is yours. Thank you very much for joining.
Thank you, Joseph. I think I would need your permission or somebody who is the admin of this call to share the slides. Otherwise, I can't share the slides. Oh, now I can. So thanks. I'll do that just in a second. I hope you can tell me if you see my slides. From the Lviv people, could you?
Yes, everything
right.
works.
Amazing. Thank you very much, Rostislav. Right. So first things first, I really enjoyed the previous talk. I'm happy that I was able to join a little earlier. So I was really fascinated to listen about hyperparameter optimization, especially when it comes to such difficult things such as fairness, which is super hard to define, right? So thanks, Denise, for this. And also, I'm very grateful to the organizers for inviting me. Yes, indeed, I'm a lecturer at the Institute of Computer Science. From the next week, I'm going to be associate professor of the artificial intelligence at the Institute of Computer Science. I'm also invited lecturer at the Ukrainian Catholic University, and also co-founder and chief science officer at Better Medicine. So we are basically building AI models for doctors. That's short as it. But I think the most important affiliation that is relevant for this talk is that I'm head of the Biomedical Computer Vision Lab. So this is the talk is going to be about projects that Biomedical Computer Vision Lab was having over the years that is centered around biomedicine, and the papers that we have been basically research that we were having over the years. So I'll give you a set of selected research projects that me and my lab, we were pursuing over the years, and I would be absolutely delighted to share as many details as I can about these projects. Before we delve into details of these research projects, I usually give a short introduction of what is this field about. And when I tell about biomedical field and research on cells and cancers, it's good to put people into context of what scale of a problem we are talking about here. And when I'm usually in the audience, and I'm very let's say I'm very sad that I can't be in the audience. I would love to be in the audience there in Lviv. I missed you guys so much. I usually ask people a question such as, how many stars do you think there are in the galaxy? And usually people don't know the answer, and I'm really glad, because that would mean something is not right. And I usually tell them it's about 200 billion stars in our observable galaxy. And then I ask them another question, which is, how many do you think we have cells in average grown human body? And that also takes quite a bit of time for people to figure out. And unless they're Googling, they don't know. And the answer is about 37 trillion cells. So it's actually trillion cells comparing to 200 billion stars. So the scale of the problem, when you think about studying cells in a human body, it's a lot. It's much bigger number when it comes to if you think about galaxies and stars. Also, comparing to stars, our bodies are a lot more intricate. They're parts, cells that communicate in so many interesting and non-trivial ways. So there are a lot of things that is happening in our bodies. So it's basically these trillions of cells that make up tissues. There are about a dozen of different tissues, and these tissues make up organs, and all that machinery is there to help us live happy and healthy lives. And as you might have followed my talk so far, this system is very complicated. It's very complicated. It's very subtle. It's very fragile as well. So there are a lot of things that can go off, that can go wrong in this system. And one of these ways how things can go wrong is that, we usually, throughout the lifespan of a human, we tend to acquire mutations in our DNA. And some of these mutations, they cause the cells to divide rapidly and uncontrollably, causing this division to invade the tissue where the cell was initially residing, and then forming this kind of lumps in the organ. These lumps tend to grow and send their messengers to other organs, practically invading other organs, the process which is called metastatic spread. And this metastatic spread essentially leads to complete demise of a person, complete seizing of life, basically killing the patient. And this is a very complicated problem that I'm illustrating. So this is a story of cancer. If some of you might know, some of you might recognize, this is how cancer is developed, and this is what happens when the patient is sick with cancer. And now I'm coming back to the computer science part of my title of my talk. So my title of my talk is about computer scientists making progress or making difference in medicine. And us as a computer scientists, we have been not really solving that sort of problems, as you can see on the right side of the slide, but we have been more thinking about other things. So for example, what Denise was just explaining, for example, we were rather successful building great classifiers for, let's say, images. We have been rather successful taking most of the control of the drivers in the cars. We have been rather successful building models that can draw or can pretend to make art. So we were pretty good as computer scientists. We were pretty good at these sort of things that you can see on the left. So as it turned out, and this is what I have figured out throughout my career, is that there is a link between those things that you can see on the left and those things that you can see on the right side. So it turns out that some of these problems that we as a computer scientists were able to solve using computer science approaches, and most likely that I'm talking about AI right now, or deep learning for that matter, we were able to solve some of these problems on the right side of the slide as well, with the same type of technology. And I'll give you a couple of examples on a higher level, and then I'll go into more specific projects as we go. So what we can start with is cellular level. So I just explained to you how cancer goes from cellular level to tissue level, and then it starts to invade the organ level. So it becomes visible at the organ level. But first thing, what happens is the cellular level. So this image that you can see on the slide is actually an image of a cancer cell line. So this specific image, these specific cells, they are cells, by the way, they are cells, and they are cells of cancer. I do not necessarily right now can tell you exactly which type of cancer it is, but this is how cancer cells look like. And what usually happens when somebody is developing a drug, let's say, that's supposed to fight cancer, is that they don't right away recruit volunteers, human volunteers, or even animal volunteers. They don't recruit anyone. In fact, what they do is that they study the way how their drug is performing on cell lines. So only on this kind of in a very artificial environment. So they add this drug to the cell line, and they see how the cell line reacts to this drug. So what they want, obviously, to see is that if it's a drug that's supposed to kill the cell line, cancer cells, then that the cells would start dying, right? And one obvious problem with this is that cells are very, very tiny, which means that counting and looking at the cell lines by eye is very, very difficult. So you can't really count all the cells. So instead, what we were doing in our lab, in collaboration with a company called Revity that produces microscopes for laboratories that study cancer cells, is that we were training very simple convolutional models to segment and detect these cells, and that would help biologists and lab practitioners to quantify and gain evidence for or against certain drugs that they experiment with. So that is that was the thing which we started with in this field. And then and there are very different ways how you can study cells. And the previous picture I showed you fluorescent images, which is just a way to illustrate, to visualize cells. And this image here is much hotter. It's called bright field images. And in bright field images, it's much more complicated to see cells. And we were able to adapt existing approaches for cell segmentation and detection, and we were still able to quantify and detect cells from these more complicated images, which were before that considered to be a junk material, was not useful at all. Now, when we move on a little bit up the ladder of the cancer progression, then we come to the pathology or tissue level. And what is happening at this level is that when people suspect having a cancer, they usually take a biopsy. Biopsy is a surgery type of procedure where they put a needle in a human and they retract a small piece of the cancer, basically of a tumor. So they take a part of the tumor, they slice it up, and they put it under the microscope, and then they study this images. So this is the image that you can see on the left side of the slide. This is how the sort of tissue looks like under the microscope. So it's a little bit higher scale image than what you just seen in the previous slide. In here, we collaborate with local hospitals and local pathology labs that usually do all these operations manually by just looking at the microscope. We help them to detect different structures onto these pathology images, and we help them to make the diagnosis. So we help them to automate the diagnosis performance. For example, with this specific image, we help to detect the structures called tubules and also count cells within the tubules, because that was a very important marker for the disease in that very specific case. By the way, this specific case is not really a cancer case. This is actually a male infertility disease, and those purple things are called tubules, and the cells inside, they indicate whether a human, a male person is there infertile or not. And then we finally come to the organ level research that we do. And this organ level research we do with a company called Better Medicine. As I said, I'm one of the co-founders of this company, and currently at present, I'm acting chief science officer of this company. And what we do with Better Medicine on the CT images, so these are called CT images, we help to build AI models. In our case, we built CNNs and some transformer models to detect and measure various organs as well as pathologies that are present on these images. So, for example, here there is a big pancreatic tumor. So we have liver, we have kidneys, and we have a pancreas. And in pancreas, we have a huge pancreatic tumor that can be detected and measured. And this is what we help doctors to do. We basically do this work for them and give them the final results for validation, for verification. That is a short summary of what I have said so far. Hopefully you were able to follow. So on a cellular level, we are helping to detect and quantify cells from microscopy images of various kinds. On the tissue level, we help pathology labs, and we work with local Estonian hospitals to help to automate this work of a pathologist, somebody who has to look through hundreds of these huge images and understand all the structures and find all the structures. So some part of their work can be automated and can be made more efficient. And finally, on the organ level, we're building deep learning models. We're training deep learning models to detect and measure and quantify tumors. That's what we do. And there are selection of several projects that I wanted to highlight that either have resulted in publications or are very close for publications, and that I think are most interesting. So the very first project, and just a reminder that we were using this kind of bright field images, and we were detecting nuclei from the bright field images. And as I probably also mentioned, is that the bright field images, so this is a bright field image, it's very hard. If you look carefully, you look long enough at this image, you might understand why is it hard. Is that there is a very low contrast, there is very little signal present, and signal noise ratio is very skewed. And although there is an advantage to these images, and the advantage is that it's very easy to acquire. It's very easy to get these images. Before the deep learning revolution, these images were considered to be junk. They were considered to be non-useful because humans could not work with these images, neither could classical algorithms, classical computer vision algorithms. And partially you can understand the complexity if you look at the overlay of the fluorescent images. So this is an overlay of the fluorescent channel with the bright field channel. So you can see the centers or nuclei of the cells, which you could not probably see on the bright field itself. But that is something that biologists want to see in their experiments. And using fluorescence is good for detecting cells, but the problem is that obviously fluorescence comes with a price. You have to pay money, you have to spend time to prepare the experiment, and the third side effect is that fluorescence kills the cells. So if you would like to study cells for long enough, then you won't be able to do that with fluorescence. So this is something you can do with bright field, but it's not something you can do with fluorescent images. Finally, so we did make it happen. We did train a model that was able to find nuclei from bright field images, something that humans could not do, but thanks to the interplay fluorescent and bright field images, we were able to train them. And now it's been deployed in production in a big company, and they offer it as a standard offering to their clients around the world. But we didn't stop there. So this was just a part of the plan. Another part of the plan was to detect the whole cell, so the full area, which is covering the cell. And that is a lot more complicated problem. So we were after something called whole cell instance segmentation, which is like a step up in a complexity ladder when it comes to detecting cells. And what we have done here was that, oh yeah, there is one part more, there is additional part in the complexity of this problem is that cell bodies, or we call them cytoplasm, they like to overlap. They are not necessarily completely isolated from other cells, so they do intersect, as you can see here on the slide, which does not necessarily happen very frequently in the natural objects world. So all these big data sets that exist for images of the natural world, you don't normally see a bicycle behind a track, while you can see one cell behind another cell. So this is where the complexity of the biomedical field comes in. So in order to accomplish this task, we actually sat down and our students, they have labeled a massive amount of this kind of cells. So they actually by hand drew the cells to collect this unprecedentedly large data set that includes the cell bodies, cell cytoplasms, which are overlapping. And then at first we trained a very classical UNet model that also worked in a standard bright field nuclei case. but it turned out that it doesn't work. And there are several obstacles or several problems with applying UNet model on these sort of images. And one of the problems is that it's not meant, the UNet, the classical UNet model, which I hope that some of you might have familiar with, is the model that is used for a lot of biomedical segmentation tasks. It's not meant for instance segmentation. At least inherently, it's not capable of doing instance segmentation. So what we have done is we have adopted the architecture. We changed the architecture in a multiple ways, but one of the most important ways is that we added 2 new branches into the model that are actually intertwined. They're not exactly isolated. They're actually interconnected. And one of these branches we call instance branch, and the other one is called mask branch. So the instance branch is what you can see on the top, is the image which is concerning itself only with the particular seeds of every cell. So it's only interested in detecting the, let's say, the cluster centroids of the cells. While the mask branch is something you can see on the bottom of the slide, is basically tasks with the segmenting the borders of every cell, but it does not care about the in particular, it is not distinguished between individual cells, but it just finds the borders of cells. Now, when we combine the two and we add some nonlinearity layers ahead of it, we actually got a very reliable and very accurate cell border prediction from the bright field images. We have developed so far, we have collected our own data set. This is the one that you can see in the middle. We have also synthesized some data set that you can see at the top row, and we have run this model on actually on a number of public data sets. And what we see across the board is that our model seems to be state-of-the-art or very close to state-of-the-art with all the other approaches that we have tested. And it's very, very promising. And this is going to be a paper. So this is called Instance-Aware UNet. And we are finalizing a paper and submitting it to ICTERI 2025. And one of the probably driving people who are driving this research is Jaroslav Pretula. He's in the past, he's a student of Ukrainian Catholic University, but hopefully also in the future. So he's a local boy. It's been a great honor working together with him. And another project related to cells is something that we often do discard, or we often ignore as scientists who work with images, is abundant meta-information available about images. So in the world of microscopy images, we always know information about the type of the cells we are working with. We know, for example, how close we are looking at the cells. This is called magnification factor. We know the microscopy type we're working with, and all these extra things that are usually discarded. They're usually ignored, and they're not really paying attention to this. So we set out to investigate whether this can be changed, whether this extra information can influence the performance of the segmentation models. And it turned out to be true. It turned out to work. And we incorporated this information through a mechanism called squeeze and excitation. This is something that is rather popular. This is a very popular way to introduce something called attention, something that has gained a lot of popularity recently because of the transformer models. But it turned out that you can do something very similar in the convolutional models as well. I'll not go into too many details because I don't have that much time to explain the details of this project. But basically, the results look very promising. You can see if you look at the top part of the slide, then I have 3 pictures there, and the one on the left is the bright field image with the ground truth annotations overlaid with this. On the middle, you can see the baseline predictions, so the predictions of the model that did not have access to the meta information. And on the right, you can see the model that had access to the meta information. And you can see that in general, it looks more reliable. So the colors are blue is the false negative, and red is the false positive. And what we can say in this is, of course, cherry-picked example. From the quantitative side, it also performing slightly better. What we also saw, if you can see on the bottom part of the slide, is that meta information can impact the way how the model actually performs, meaning that it can actually tell the model to change its behavior. And you can see that on the, again, on the bottom of the slide from the very left, you can see that this is the huge kidney tumor, and its dashed line represents the borders of this kidney tumor. On the next image to the right, you can see in green, this is what the model produces for the mask of the kidney tumor. If you say model that the tumor is large, then the next image shows the output of the model if the meta information indicates that the tumor is medium size. And finally, the last rightmost picture shows the prediction of the model if meta information indicates small tumor. So this research has been published at the workshop at MICAI. It's one of the top medical imaging conferences. And actually another maybe a small compliment to Ukrainian Catholic University is that one of the main authors was Yaroslav Plutenko, who was my student at Ukrainian Catholic University. That was the cell level. We have a lot more projects related to cells, but because I want to show you all the things we have done, we are moving on to the tissue level. And in the tissue level, one of the most complicated problems is the sparsity of the annotation. So because the tissue slides, if you have ever seen pathology images, they are huge. There are thousands, or let's say hundreds, or even dozens of hundreds of pixels in diameter and size. They're very big, and they are very dense in terms of information. So it takes a lot of time to densely annotate those images. And what usually happens is that pathologists they skip some cells when they annotate those images. So for example, you can see that some boxes are there around some cells, but there are many boxes that are not been given by the pathologist. And this is something that usually confuses the model a lot. And the last image here is the annotation that we would love to expect. This is something that we would want to have. In reality, the middle picture is what we usually have in the pathology world. Now, what we have done to remedy this problem is something called generalized background recalibration loss. So in a nutshell, we are telling the model that when it produces false positives, so it's not, let's say, when it thinks that or let's say when it predicts a cell, but there is no cell in the ground truth, this is usually referred to as false positive, and it's a penalty that the model takes in as a feedback, as a gradient descent in terms of gradient descent. But the generalized background recalibration loss, it's actually discounting this false positive error. It says that if the model is predicting very confidently false positive, then I take it as a real signal, because I know that there are a lot of cells that do not have annotations. So if the model is very confident that there is a cell, it must mean that indeed the cell is there. And it turned out to work, as you can imagine. So the left side of the slide is the ground truth. So all the boxes are the boxes from the pathologists, and we asked them to be very, very precise and to annotate as dense as they can. So they did what they could. And in the middle, you can see the work of the baseline model. So the model that was not trained with recalibration loss that we have proposed. And the rightmost column is the column where you can see the results of the model that had this loss in place. And you can see that the model actually performs much better. It generates a lot more boxes. But of course, this is again the visual example. We have, you can check out the paper. This was published as a tiny contribution into ICLEAR 2023. You can find the paper and check out the results in more detail. Finally, we have moved on to better medicine. So this is where we perform organ level research. At Better Medicine, they're taking this kind of full body approach, meaning that they want to build deep learning models for segmenting tumors in all the organs inside the abdominal cavity. This is what you can see is illustrated on the slide. And at the moment, what happens is that Better Medicine has lungs and kidney models in a clinical validation and certification, and liver and pancreas models as a proof of concept models. These are very early results for these two models. Again, as I said, for the kidney model has been rather successfully validated. And the lung model, so the model that is detecting tumors in the lungs, it's been going through the validation process. Now, a little bit more about the kidney model. We call it renal vision. And what it does, it essentially performs semantic segmentation into three classes. And these are kidney tissue, a tumor, or a malignant finding, and the benign finding. So these are the three classes which we are detecting. And once this has been done, once the segmentation has been performed, the next step is to measure and report these findings to the radiologist. So we tell the radiologist, hey, we found this many malignant lesions, this many benign lesions, and this is their sizes. Because this information is very important for the clinical decision making. As you can imagine, medical community will not accept anything that is not proven to work reliably, and therefore we have run and executed a special clinical investigation in which we have validated this model. In this investigation, we employed 6 local radiologists who have examined 200 CT scans, both with the help of our model as well as without our model. There was a washout period involved so that we can actually have more data for the study. And we have randomized the study. So basically radiologists didn't have the same order of the study, so they couldn't really communicate with each other. So all sorts of biases have been taken into account. And what this study has shown is that we increase the productivity of the radiologist by, on average, 33% and up to 52%. So this is the speed up of the radiology workflow. Second thing, what we have shown is that we slightly increase the patient level sensitivity, which means that they are capable of finding people with kidney cancer and more accurately. And finally, we have increased the interradiologist agreement rates, which means that they would agree more frequently with each other about the correct diagnosis, about whether a kidney lesion is malignant or benign. So this study is being finalized and will be soon submitted to the journal, aiming at Nature Oncology. But of course, the editors will always have their say, and of course there might be changes in that plan. Why we do measuring lesions is because we would like to figure out the dynamics of the cancer. And what happens when people get diagnosed with cancer is that they are usually invited for a follow-up scan. They come for the follow-up scan, and then the radiologist has to examine both the original scan and the follow-up scan. And this is where our kidney model would come in. It would segment both scans. It would identify all the lesions in those scans. And then what we do is we do something called alignment. So we register the 2 scans so that we can understand which scans come from how to track the lesions. And once we align them, once we register the scan, we can quantify those lesions, we understand their volume, area, and diameter, and then we produce a comprehensive report for the radiologists for their decision-making so that they can use it for the decision-making. The problem is that it's much harder to do than it sounds. So this registration is very complicated because people are changing over time. They change their posture, they gain weight, they lose weight. and therefore registration is very complicated. So recently we have applied and received funding for building an AI-based approach for estimating something called a deformation matrix, where we built basically a tensor that helps us to understand which voxels from first scan correspond to voxels in the second scan. By the way, I wanted to ask if I have a few more minutes, or is it that I have to cut my presentation short? I have like a few slides left.
Yeah, please continue. Please continue. We started a bit later. So.
Yeah. I just wanted to make sure that that's all fine. Right. So that is the story about the project related to dynamics assessment. So with this, what we do is we track lesions over time. And with this deformation matrix, we understand how lesion has changed in the past. How has it been in the past, and what is the current state of the lesion? Finally, two last projects, which I think are very, very interesting and very important. And one project is related to something called counterfactual in painting. So I'll try to explain. Here, what you can see on the left side of the slide is the CT scan with the kidney cancer. So there is a kidney cancer in the circle, in the red circle. Now, just a bit of a context here that one of the most expensive and one of the most complicated parts of being in biomedical research is data annotation and data in general. There is a lack of data, and there is a even harsher lack of annotated data. And data annotation in this field is very problematic because it involves hiring very highly skilled professionals who earn a lot of money. So you have to pay them a lot of money, and it also takes a lot of time. So instead of, so basically training full-fledged deep learning models that require pixel level or voxel level annotations is very complicated. So instead, what we're trying to do, weakly supervised approaches. So this is an example of the weakly supervised approach where we say, okay, what if we just say that the scan contains kidney cancer? Can we make use of that information? Because this information is relatively easy to come by. So what we do here is we train a classifier that can tell whether the scan contains kidney cancer or it contains a healthy individual. So you train that kind of classifier. Now you feed the prediction of this classifier into another model, which is called Generative Adversarial Network, GAN model. And the purpose of this model is to generate an alternative version of the scan such that the classifier will be tricked. It will not be able to predict the correct class from this alternative version of the scan. And it turned out that the only way this can happen is to GAN to generate an image without kidney cancer. So it generates, as you can see here, this is an image with a second image with a circle. It generates a scan where there is basically a hole instead of kidney cancer, right? So when you subtract the original image from this generated image, you get the mask of the tumor. This is where the tumor is located on the scan. And this is fascinating because if you think about it, we just started with an idea that I'll just tell you that there is somewhere a kidney cancer on the scan, but I'm not telling you where. And then the whole approach learns to find where on the image is the kidney cancer. And I warn you that the kidney cancer is a very complicated thing. It can take any shape or it can take any morphology. So it's very complicated. There are a couple of examples how this approach works. In the red contour on the left is the input image, the middle image is the counterfactual, and the difference map is what we basically refer to as the location information, is what you can see on the right. And there is another example of the same sort. And the final project that I wanted to discuss, and very quickly though, is using latent diffusion to generate more conditionally labeled data. So again, the same problem. which we are facing is the lack of annotated data in the field. And one way to get more annotated data is generate it. And the way how we generate this annotated data is through conditional generation. So we provide a mask of a tumor that we sample from the public available data sets. We add it to the scan of the healthy person, because most of the people that come and get their CT scans done in the hospital do not have anything extraordinary on the scan. We put it on the scan, and then we add it as an input to the latent diffusion model that is producing a scan, which now in this place where there was a mask, it produces a kidney cancer. And now what we have is we have a scan from a hospital with a person that has kidney cancer in a place where we wanted it to be. And this is obviously without any people being harmed in the process, right? Nobody got their kidney cancer, which is great. But instead we have a scan where we have a mask and we have a corresponding image. And we can now train our AI models. We can train our UNet models to detect these. And these are just a couple of examples here. So again, on the left, you can see the input image with the white mask that is just being placed there as a placeholder for a model to fill it in. The middle column shows the kidney cancer being generated by the model, and the right image shows you the actual scan, so you can see what was there underneath the mask. So there was no kidney cancer, as you can see. The same is for the other example. This one is a bit more pronounced, and you can see that the model was able to generate kidney cancer there. We could do a very similar thing with the basically generating healthy examples instead of, so if you have a person with the kidney cancer, you can actually generate healthy example by just occluding kidneys. This was a very interesting finding of that project as well. And finally, because I don't want to test the limits of my time availability, I'm wrapping it up, just saying that what we have talked so far is that on my lab, we are doing research in biomedical imaging field. We work with images of cells where we detect cells, we quantify cells. We work with tissue images where we help pathologists to automate parts of their workflow. And finally, we build deep learning models to detect and measure and monitor tumors over time in CTs. So that's all from my side, and this is my lab. The numbers in the bottom are already outdated, and this is the company. So if you wanted to see, these are the real people behind the slides. Thank you.
Thank you very much, Dmytro. It's a very impressive work.
Thank you.
Very nice and very nice presentation too. Yeah, thank you. So we are a bit over time, but let's still, have a few questions. But I will pass to Rostislav if there are any questions from the room.
Yes, thank you, Josef. Yeah, so thank you for your talk. And I have a question about, you showed models that you built and you tested them. It seems they quite well. But for me, it's interesting, what types of tests should you pass to push your model to production, especially in such field as like healthcare? I know it's quite hard, it's quite long to explain, but maybe you can provide some grasp what types of requirements they put to you to push your model to production.
Thank you. I think it was Denise. No, if I'm mistaken, who gave a previous talk, but maybe I'm wrong. Thank you for a question, by the way, because I can't see you that well. I'm only seeing like a bigger picture. I don't see the details, but indeed, the path is very long and full of horrors. I can tell you that. So one of the probably most important requirements is that company that wants to deploy these models in real hospitals and actually treat, or let's say in our case, diagnose real patients, they have to make a clinical investigation where they have to show with real data, with the hard data, that indeed their whatever system that they are trying to implement works. So because we are not a class 3 device, so we are not interfering with human liquids or tissues, then we didn't have to do it in multiple trials. We did one study with a real radiologist in a real clinic. So this is what I'm showing here. This was done in real clinic. It was not in a way fake or made up. So this was in a way real people who work in a real hospital. And the data that you collect, it has to be convincing enough. So it has to be significant, obviously, with all the tests being significant. You have to show that you have avoided all the major biases, such as what about your gender distribution? What about your so all the fairness questions that if I understand correctly, who's asking a question, all the fairness questions are very, very relevant here. So for example, we have had half of our population of the scans coming from females, half of them coming from males. There was an age match. All these things have to be taken into account in order to convince multiple reviewers that indeed you have done everything correctly. Before that, you have to go through something called a QMS. This is a quality management system where you show that whatever you do in a company is of a good quality. Then there are ISO standards, which is international standards of software development, AI development, all these things. These are in parallel to the clinical investigation. So clinical investigation is one hand, and there are all this ISO, QMS. Then there is a final audit of the committee, which looks at all the data that you have provided, and they give you lots of questions. Then I think we have spent like half a year, I think, only answering questions. And finally, we are right now at somewhere close to getting the CE mark.
Thank you.
Thank you. Any more questions? Rostislav, please.
Thank you. So I have a question related to if you follow up performance or progress, how your technical approaches influence doctors, because, for example, in my understanding, it can raise like technical bias. If I'm a doctor and I have help from a technology, it may later influence my decisions. Do you follow such metrics or something like this?
Yeah. So when you convince reviewers that indeed it's gonna be fine, trust me, it's gonna work, then they always say, no, we don't trust you. It's not gonna work. They ask you all these questions. So they ask you, what about biasing radiologists and that and this. So indeed, we have seen it. Even in the study, we have seen that some radiologists trust the model. That's it. And actually, it's a little bit something that shows through this graph. It's a rate of a speed up that basically from the start of the study all the way until the end of the study. And you can see that the rate of the speed up is increasing. And one reason why it is increasing is that more and more radiologists started to trust the model because they saw that the model is making sense. And one way to mitigate this over trust or the technical bias, as you called it, is to inform. Is what we have in our intended use in all the documentation is to say that you are the final responsible. You are the person to be responsible if something happens. Then the other thing which we do is we do put all these warnings that, are you sure? Have you looked? Have you checked? Is it all right? Is it okay? And the final thing is that, of course, we are relying on radiologists to catch up, to prevent bad things from happening. But what we have to do ourselves is to ensure that the model is performing the best possible way. And even if the pilot is drunk, the plane should land safely.
Thank you. Any other questions here? I have a small question. You showed 2D segmentation of malignant tumor, but isn't also interesting to build a 3D segmentation? It may give more information for doctors about the size and location.
You're absolutely up to the point. And actually, all the segmentations I showed are 3D, all of them. And the reason is that the problem is that slices are very similar. nearby slices are very similar. They're almost 99% similar sometimes, depending how thick the slice is. And if basically if you train a 2D model, then for each slice for a 2D model is like a new image. And this is obviously not the case. It has to take into account all the surrounding. And that's what 3D models are meant for. And this is another problem because 3D models are harder to train. And but that's what we do. We do 3D.
Yeah, thank you. Yes, we have another question.
Thank you for the talk, for the great talk. One small question. So in one instance, you generated images using a deep learning model, I think, and then so images which do so images where it is simulated basically that the cancer is there, and then you trained another model to distinguish between the between images where the cancer is there and where it is not there. Did you maybe check whether this behaves in the same, so whether the generated images are realistic enough so that the model trained on this artificial data will be reliable then?
Right. So I expected this question. Thank you. Every time I speak about these models, I get this or different versions of this question. Basically, the question is, are you sure? The answer is no, we are not sure. So far, so we are still in a progress with this research, so we are not sure. We haven't implemented anything that would affect patients, and in this case, it's a pure research yet. And so far, I was able to trick radiologists into believing that artificial stuff is actually real. So I take that as one hint that indeed the model has generated something realistic. But what you're suggesting is completely right. And what we are doing right now is actually testing what the model's actual behavior is going to be like. Would it help? Would it introduce more problems? You're absolutely right. There are a lot of questions that needs to be answered, and this is research in progress.
Thank you.
I have like another question. Have you tried any uncertainty quantification methods for your models?
We have not, and I regret this deeply because we should and we will. Well, as long as I am a CSO there, we will for sure. And the reason is very clear. We need to give the estimate. We need to give some kind of basic uncertainty estimate. At the moment, we don't, and that's a big issue.
Yeah, models sometimes can show when they don't know what it's important, it seems.
Yes, I cannot tell you how much I think this will improve the user interaction. Absolutely.
Yeah, thank you. There seem to be no further questions. Joseph, do you have any?
Yeah, or maybe just a quick comment on this in the image which you show. I think the good news is that you can actually quantify, what really matters in this performance on kind of held out test data. So I think if you can show that the generated images help that in a kind of a, unbiased and also kind of non-leaked way, I think, that was going to be the real proof. And the synthetic data, it's really storming different fields as well. It's being used in autonomous driving, which is also safety critical domain. There, of course, there is much more training data available in general because, it's, I guess, easier to capture. But I think there is in the end, like the real proof is the real test results which you can get. So I think.
Exactly. That's what I keep telling to all the people that you have no idea how many people ask me questions about the synthetic data. First of all, it's very trending, and therefore they're like, they know about it. And yes, absolutely. I think for me, the final indicator is, does the model work? Does it work on all the scans equally good? Yes. And then I'll start looking into figuring out what exactly is wrong and what are the typical problems that it's having. For me, the realism of the generated tumors is a secondary thing, because if at the end of the day, if the model is doing things on the real data or the real patients, then that's the most important indicator. But of course, we are using public data a lot for these sort of things as well.
And it's true that in other domains like robotics, even non-realistic data help the classification, like the output performance, like this domain randomization work with arbitrary textures. I think so. There is as long as the real data is within the kind of, the manifold of the generated, it's okay. So I will sort of not delay this further. Thank you very much for this wonderful presentation and very interesting discussion. So thank you again. Thank you.
Thank you very much. Thank you, audience, as well. And, Slava Ukraini.
Thank you, Dmytro. And with this, let's close this exciting session. I will thank all the speakers again for very nice presentations. Rostislav, do you want to have any final words?
Yeah, I would like also to thank all the speakers of this afternoon or second session in the afternoon, to Michal, Denis, and Dmytro. And also I would like to thank the main driving force behind this event. Thank you, Vadim, for suggesting these form of participation, and thank you, Joseph, for actually making it done.
Yeah, I just have to say a few words for the closure of this company. Thank you, Joseph. Thank you, Rostislav, just for making this wonderful event, sub-event at ICTERI, true and very useful to the public. Enough audience here, everybody's happy. So thank you very much. I do think that lightning talks at ICTERI have a real future. Very good work.
Thank you. Thank you, Vadim.
So bye-bye and see you next time.
Thank you very much. See you next time. Hopefully in person.
In person in Rio.
Yes.